ultrafiltration, blood is filtered at the start of the nephrons. The long tubules, along with bundles of capillaries, is where the blood is filtered, and these are called nephrons. There are thousands of nephrons in each kidney. Blood from the renal artery enters smaller arterioles in the cortex. Each arteriole splits into a structure called the glomerus, which is a bundle of capillaries looped inside a hollow ball called a renal capsule. The blood in the glomerus is under really high pressure. Ultrafiltration takes place in the glomerus. The arteriole that takes blood into each glomerus is called afferent arteriole, and the arteriole that takes the filtered blood away from the glomerus is called the efferent arteriole. The efferent arteriole is smaller in diameter than the afferent arteriole, so the blood in the glomerus is under high pressure. The high pressure forces the liquid and small molecules in the blood out of the capillary and into the renal capsule. The liquid and small molecules pass through 3 layers to get into the renal capsule and enter the nephron tubules. The capillary wall, a membrane called the basement membrane, and the epithelium of the renal capsule. Larger molecules, i.e. proteins and blood cells, cannot pass through and they stay in the blood because they're useful for us to keep. The liquid and small molecules, now called filtrate, pass along the rest of the nephron, and useful substances are reabsorbed along the way. The filtrate flows into the collecting duct and passes out of the kidney along the ureter. So during this whole process, the waste products are removed from there, because if we leave urea and waste products, they're obviously going to build up, and it'll be quite toxic for a human being. Useful substances are reabsorbed along the nephron tubules. Selective reabsorption takes place as the filtrate flows along the proximal convoluted tubule, which is before the loop of Henle. The distal convoluted tubule is also where selective reabsorption takes place. So useful substances leave the tubules of the nephrons and enter the capillary network that's wrapped around them. The epithelium of the wall of the PCT, the proximal convoluted tubule, I'm going to call it PCT from now on, has microvilli to provide a large surface area for reabsorption. The microvilli are like, they have sort of, it's a bit like folded sort of membrane, so it's got a bigger surface area. So this helps obviously for the adaptation of the folds, provides a large surface area for the reabsorption. So useful solutes like glucose, amino acids and vitamins. Some salts are reabsorbed along the PCT by active transport and facilitated diffusion. Some urea is also absorbed by diffusion, but not a huge amount really. Water enters the blood by osmosis because the water potential of the blood is lower than that of the filtrate. Water is reabsorbed from the loop of Henle, DCD, and the collecting duct, which is called countercurrent multiplier mechanism. The filtrate that remains in the urine is passed along to the ureter and then is taken out in the bladder, obviously. So the urine normally contains water and dissolved salts, urea, and some substances such as excess hormones and excess vitamins, but you've got to make sure that you know that it doesn't usually contain proteins, blood cells, because obviously they're too big, glucose, amino acids, and vitamins, because they're normally reabsorbed back into the bloodstream, but obviously excess vitamins are released there, but there's not always a huge amount. The countercurrent multiplier mechanism is in the loop of Henle. So it's got the descending limb and the ascending limb. They help set up the countercurrent multiplier mechanism. I'm just going to call it the TMM from now on. It helps reabsorb water back into the blood. So basically it starts on the ascending limb of the loop of Henle, and here they pump out Na plus and Cl minus, sodium and chloride if you don't know that, and these are actively pumped out into the medulla. So, and the ascending limb is impermeable to water, so the water stays inside. This creates the low water potential of the medulla because there's a lot of ions in there. So because there's a lower water potential in the medulla than in the descending limb, the water moves out of the descending limb into the medulla by osmosis. This makes the filtrate more concentrated, so more ions, but the ions can't diffuse out because it's not permeable to them. The water in the medulla is reabsorbed into the blood through the capillary network, near the bottom of the ascending limb, so where it just changes from the descending to the ascending, so the loop sort of bit. Na plus and Cl ions diffuse out of the medulla, which lowers the water potential even more. The ascending limb obviously is impermeable to water, so it stays there. These first three stages, they increase the ion concentration in the medulla, which lowers the water potential. So this causes water to move out the collecting duct by osmosis. So the water is in the DCT, but it then moves out by osmosis when it goes down the collecting duct and back into the medulla. So this actually makes sure that water isn't in the urine. So if we don't have enough water, more water will be diffused out by osmosis because we don't have enough in the body, basically. And that's basically summing up the countercurrent multiplier mechanism and the filtration of waste products in the nephrons. I hope you help.